print off. I'll send out a notice on Sunday when it's ready to go, and you'll print it out. And everybody, in the past when I had more time, it seemed, I gave everybody an entirely different exam from everybody else, and that requires much more grading on my effort on my part. So I've gone through various modifications, and in 431 I haven't done that nearly as much. I don't mind if you work in groups. However, although I cannot enforce it, obviously I expect you to do your own work. So discussing concepts and trying to figure out, information, I think is part of the learning process and is really helpful. As an instructor, it's really fun to listen to you do that and argue over points and, not points, but I'll argue over facts and how to interpret them and utilize them. So that part I encourage and I think is great for you to do. But what I certainly don't think is fair is to ask to see somebody's test and then, copy their answers or that type of thing. So it makes people feel uncomfortable when you do that. But of course, ultimately, it's up to the person to decide whether or not they're going to give your exam for them to look at. Okay? So don't put them in that spot. Certainly discuss questions and concepts. Say I found this information on this site that they can go and read on their own. But ultimately, a take-home test is a learning experience. Okay? There's not as much. When we did it last semester, we had primarily the autonomic nervous system, and that was a ton of details and drugs and all of that. More so with urinary than respiratory, not as much as autonomic. We have the concepts of diuretics and mineral control and stuff like that. So that's why I put that on a take-home, because there's a lot of complicated physiology that I want you to be able to work with, but not necessarily memorize. So, back to here. You'll notice that there are no A, B, C, Ds. Every question has 4 or sometimes 2 answers and available. And this is a mark all that applies. All right? So you get a quarter or a half point, and I don't remember how many total questions I have here. It was, I believe, a quarter point when we did it for the nervous system. So this would be worth 1 point. If all the answers were correct and you only marked 2 of them, you lost a half point for not marking the 2 that were correct. Okay, so you get credit for if an answer is not correct and you don't mark it, you get credit for recognizing that it wasn't correct. So you don't have to have the entire question perfect to be able to get credit from that. So you can get partial credit. Now, if you just go through and mark every answer, if you don't mark anything, you would get some points because not every question, every answer isn't going to be correct. All right, but you would get marked off, obviously, for questions that were not correct. So you're going to mark through. If only one answer here is correct, you would just mark that one answer. So any combination can occur. You may have the first 2 are correct and the last one is correct, or just the middle 2, or none of them may be correct, or all of them may be correct. Okay? So this involves, I grade each of them by hand. You're not going to use a Scantron, so you're going to mark directly on the test. And it's helpful. I didn't have too many instances last semester, but it's helpful because the internet has such a variety of information. Some is accurate and some is not, that you keep a log of where you find, like the website, or maybe a screen copy of that page that you can keep as a file. Okay? And then rather than print everything off, you can just send it to me if we have disagreements on what the answers are. So let's look at the very end. This is what again, this is last semester, so there may be modifications. So there's a diagram here about the osmolality and how the kidney functions to conserve or eliminate water. Here's a chart about what happens at different regions of the nephron that you would place in this table type of thing. Here's a sequencing of where molecules of ions, sodium ions or glucose molecules might go, that type of thing. Okay? So this is due, let me look up our calendar. and I'll make a final decision on that. It's not due when we get back from spring break, as some bad rumors have been going around, because we only have a day's lecture on that. So let's look up our schedule. So your exam is on the 29th. I will have this due the, let's see, yeah, we can grade it. I'll have this due on, by Saturday, the 3rd. Okay, so by midnight, Saturday, May 3. So we have spring break, and then we have a week. It says lecture exam here. We will be having a in-class lecture exam because that's your take-home. So we'll just be having the lab exam, and then we'll start lecture on the digestive system, and then your take-home will be due Saturday night by midnight. Okay? You can either email it to me or you can turn it in class on Friday. If you don't turn it in class on Friday, you can just scan it and email it to me, and I'll be happy to print it off, but that would be the last date. Okay? All right. As far as how people do on take-homes are not easy, at least mine are not, because they are a mechanism by which I can direct you to learn material that we may not have covered in class to that level of detail, and but it's for that purpose. So you probably would spend more time on the take-home than you might otherwise study. Grades, people that tend to have grades in the 50s and 60s on the lecture exam tend to move up to 75 to 85 because they're got access to information. People who tend to get 98 to 105 on a lecture exam tend to come down to about 97. Okay, so what I normally see in my span of grades tends to do this. All right, because I don't typically have extra credit on a take-home, so the 105 isn't available. But that's kind of how the span goes. Okay, so we're looking at urinary system, and on page 3 is a series of functions of the urinary system, primarily the kidneys, which may have a greater variety than you're used to. What have we talked about the urinary system so far in reference to previous units?
Filtration.
Okay, filtration, certainly a mechanism of getting rid of waste products. Anybody know, although we haven't discussed it, what type of waste products? The kidneys. Well, we get urea, so I'm looking more for nitrogenous waste products. So urea, ammonia ions, some degree more solid uric acid, because we have the fluid to dilute that. So ions, certainly that's one of the mechanisms we're going to look at in our last lecture for how we control acid-base balance with hydrogen ions and reabsorption of bicarbonate ions. Because of the fluid, it makes sense that it's a volume control, a blood pressure and blood volume control. So we looked at hormones very briefly, aldosterone and ADH, that help to regulate urine output as the mechanisms for controlling blood pressure. And the reverse is true. Blood pressure needs to be regulated because it's our filtration force. So if blood pressure is too low, filtration can't occur adequately, and if it's too high, it can damage the filtration membrane. In addition, remember that kidneys are an endocrine organ. We talked about erythropoietin with the red blood cell synthesis. It is also important in additional ions such as calcium and magnesium and potassium. So people who are on a renal diet have to be aware of foods that are high or low in those particular components. And so as we go through, we're going to highlight some of these functions and where in the kidney they take place. By specifically where along the nephron tubule they take place. So let's look at the anatomical location. Actually, we'll look at that and then we'll move into embryo. So here we're looking at a cadaver. This structure here on the cadaver's left is the aorta. The inferior vena cava is to the right and anterior, so the renal vein here is going to be crossing over the renal artery. You'll notice that the left kidney is a little bit higher than the right one. Anybody have any idea why? I know we haven't done digestive system yet, but
I was going to say maybe the liver.
yeah, exactly. The liver is much larger than the spleen. It's the other dark purple reddish organ. You can see it kind of part of it up there. And so we have the the liver on the right side pushing down the right kidney about 1 inch to 1.5 inches lower than the left kidney. Notice our renal vein here on the left. Remember, it drained directly into the gonadal vein, drains directly into the renal vein. Now, what membrane is going to cover the kidneys? Yeah, it starts with one of those P's. Not visceral. Parietal pleura. So the kidney is considered to be a retroperitoneal structure. It develops from the mesoderm of the posterior abdominal wall and is behind the peritoneum. It's a primary structure. So looking at the embryonic development helps explain that a bit. And we've already covered these 3 stages of the kidney development. What was the significance, if you remember, of the pronephros? Remember, this is unusual. No other organ in the body appears and then disappears, and another one shows up, and then that disappears, and then finally the primary functional kidney shows up. Anybody remember some of the significance about the pronephros as far as its function? Does it filtrate? Does it make urine or not? No, it's the only one that doesn't. So if it doesn't do any filtration, anybody remember what its role is? We talked about it with the development of the reproductive system. The key word I'm looking for starts with an I. Think about retina and the lens of the eye, the outgrowth of the optic nerve and the invagination of epithelial tissue forming the lens. What was the influence between those 2 structures? Anybody think of the word I'm looking for? We also use that same word when we try to get a woman to have her baby early.
Induction.
Yes, induction. So the tissue of the pronephros induces or encourages, influences the development of the mesonephros as well as the ovaries or the testes, the gonads. So if we look here at a posterior frontal section, so from the posterior view, the pronephros, remember, starts up very high, and before it's completely gone, the mesonephros appears. And the mesonephros is what we talked about. The mesonephric tubule was the male reproductive pathway, and the mesonephric ducts form the efferent ductules, link back. The mesonephros actually will start to filtrate blood and creates a filtrate or urine. And then finally that disappears, leaving just the mesonephric duct in the male, completely disappears in the female except for maybe some remnant subductuals. And the metanephros grows upward from the bladder, if you remember that clay animation that I showed back in the endocrine system. So the metanephros is actually our functional kidney. But even though it starts down here and goes upward, it ends up in a high location. So it's going to be present in the posterior abdominal wall, just along the origin of the diaphragm. So it's developing from the mesodermal structures here of the embryo, which is interesting because it has epithelial tissue. Yes, Jim? All right, so you can see a sagittal diagram here in the upper left of the pronephros gradually disappearing as the mesonephros appears, and then the metanephros down here. You would expect with that development of the metanephros that our kidneys would be found in the pelvic region. But they rise up to be found just below the diaphragm, as I indicated earlier. So in this structure right here, you can start to see it up there with the mesonephros. We start to see the intimate association between epithelial tissues and blood vessels. The epithelial tissue structure is going to form, in our true kidney, the nephron. That is a tube, in places highly coiled, but just a single tube, across which we have the filtration, and within which we gradually modify the content of the blood. So the fluid of the blood and small amino acids, ions, will cross from the blood into this tubule. And then we modify the contents by reabsorbing most of that back. And we'll talk about that when we come back after spring break in the formation of urine. All right, but we're not very picky about what we absorb. It's small enough to cross the filtration membrane, it ends up in the tubule. So I often tell a story at the beginning of the formation filtration lecture. It's a made-up story. It's a parable, if you will, or an analogy. So when my kids were little, well, their bedrooms are all on the opposite end of the house of the master bedroom, and there's a long hallway that goes down by their bedrooms. My husband calls it the Hall of Weasels, and they're supposed to keep it up. And my sister-in-law sent me 500 Lego blocks organized by color, much to my dismay. Anytime I found them on the floor, I didn't put them back in the box, I threw them away. Because they appeared everywhere, like dog hair. You find it everywhere. And so, the kids would drop a pencil, they'd drop, a Game Boy or something on the floor, and you'd be surprised how many weeks would go by that they would just walk up and down the hallway and step over it and keep on going. So let's say one day I'm cleaning up the house, my mother-in-law is showing up or something, and I'm tired of all this stuff. So I get a big box, and anything that fits in the box, except computers, something I've paid for, it goes in there. Toys, clothes, baby dolls, whatever, goes into this box. Anything that fits. That's the filtration that takes place here. Anything that's small enough to cross the filtration membrane. And I put it out in the garage. Take to Goodwill or the dump. And when the kids come home from school, all of a sudden, all the stuff that they've been stepping over for weeks, they need right away. Because it's not right where they saw it on the floor 2 weeks ago, or that morning. And I say, well, it's out in the box in the garage. If you need something, you'll have to go out there and get it. So they go out and they selectively take out of the box what they want to play with that day. Reabsorption. Okay? That takes place along this tubule. So if we're not diabetic, we reabsorb all of our glucose that's been small enough to filter across. We reabsorb 98% of our water, okay, our sodium. And so, but I continue to find things on the floor. So since I haven't had time to take it to the dump, I keep making trips out to the garage to add more stuff to the box. So the kidneys continue to add material to the tubule, primarily hydrogen ions, the metabolites of antibiotics or drugs that the liver has broken down. We call that secretion. All of this time, we're not dealing with urine yet. We're just dealing with a liquid called a filtrate. And until it's finally been concentrated and is ready to enter transport tubes, that's when we finally call it urine. Okay? So that's the process that we'll be talking about when we get back from spring break. This is just other slides showing the changes from mesonephros to the actual kidney tubule. So here's what we mean by retroperitoneum. Here's the ventral abdominal cavity. And instead of lining it with a serous membrane called the pleura or the pericardium, it's lined with a serous membrane called the peritoneum. When we get to the digestive system, we'll talk about how the kidneys grow into that and are covered with visceral peritoneum, and how the stomach is suspended by a mesentery. But here we have the kidneys on either side of the vertebral bodies, and they are retroperitoneum. They are held in place by renal fascia, adipose tissue. Now, if somebody needed a kidney transplant, all right, what would be the best access? Would you want to go through the ventral abdominal cavity and make an incision here? No, because then you would be piercing that twice and introducing the risk of bacteria into the peritoneal cavity, especially if there happens to be a nick of the colon or something. So you want to come right through the back, because that's where we have our rector spinae and muscles that help to rotate and stabilize the vertebral column. So there are some modifications, but most of the incisions are lateral, all right? So just under the ribs in this area right here to provide minimal damage to posterior wall muscles and gain access to the kidneys without going through the peritoneum. It's protected by skeletal muscles, primarily two. We don't have a lot of bony tissue in that area, so this is a model, our short skeletal model, and protected by the psoas major. That's one of our strong hip flexors, and quadratus lumborum. That goes from iliac crest to the lower ribs. So remember in Bio 430, those of you that had me, we talked about some people who, trying to have a smaller waistline, will have their vertebral ribs, their floating ribs removed. That's the only bony protection that the kidneys have. Right? So if someone gets kicked in the back by a horse or by a person, and those ribs break, those can lacerate the kidneys, but otherwise they are a protective component.
Psoas major and
Pardon? Psoas major and quadratus lumborum. That should be listed on the top of page 4.
Does anybody do that anymore? Yeah, I thought you did.
I'm not. I hope not.
I don't have mine, but it's from I had spinal surgery, and so they took out 3 of my ribs.
On one side.
Yeah. But I have pain there from it feels like from it.
From the surgery or from the
I don't
lack
know.
of
It feels
support?
like poking. I just had the surgery like a year and a half ago, and so the doctor said that sometimes it's like from breathing.
So when the diaphragm is.
Yeah, it like pokes it.
So what did they do with the diaphragm muscles that would have attached, had their origin from
I
those
have
ribs?
no idea. Because I had bacteria on my spinal cord, and so it got into other parts of the bone, and so they had to remove other parts of the bone too. And then they used the rib, one of the ribs
To
as
graft?
like to fuse the, yeah. So I don't know. But it's very hollow right there. Like I have this weird like indent on my.
I'm assuming they would have attached it to ribs above, so that would limit your thoracic cavity.
Yeah, because when I breathe in, it's like.
It's not going to be as
Yeah, I feel
expansive type.
Like
Yeah.
it stops where I can't
Yeah.
really weird.
There might, part of that might also be adhesions, because anytime you have surgery
Yeah.
around the peritoneal or pleura, the simple squamous, a lot of collagen fibers
It
tend
feels
to
like
form
a stitch
adhesions.
almost, like you're getting a stitch in there. It's really weird. It's
Um,
just several nerves though.
Okay. So is that every time you breathe, or just when you're breathing deeply?
It's only if I'm breathing like really deep, like if I run on the treadmill or something, then it really is, you know, like I have to sit down and really think about breathing. It's really weird, like you have to think about
Actually,
it.
about the. Because it wouldn't be just the ribs, it would be the intercostals and the nerves for those associated with that. We don't have time for this, but this is a video I'll put a link up to on surgery for a kidney transplant. All right, so when we look at a kidney, it's a rather simple organ anatomically, okay? For our lab exam on the kidney, we really are looking at kidney, ureter, bladder, and urethra. Those are the structures. So the nephron is nephron and collecting tubule are the two tubules. The nephron has several parts, all right? But functionally, there's a little bit more to the kidney than there is anatomically. It's divided, as you can see, it's kind of shaped like a lymph node. Has the same outer convex appearance and an inner concave at the hilus or hilum. And we don't have afferent lymphatics, obviously, but we have a capsule, we have a cortex, we have a, here's the cortex and a medulla. And this area here is known as the sinus and pelvis. So essentially what happens is we have blood coming into the kidney, sending branches out to the outermost cortex. Out here is where filtration occurs and where we reabsorb and secrete. And then some of that filtrate comes down into the medulla and back out again, and a process to set up a concentration sink, a salty gradient. And then finally the filtrate leaves, it passes through that salty gradient. If your body needs to keep fluid, the salt pulls it out, and then it enters into the minor calyx here. This is the first time we actually could call it a urine fluid. Everywhere in the medulla or in the cortex, it's going to be called filtrate. So let's go through these structures. And I wish I had a higher board. You know what I'm going to do? Turn this off and diagram on the board for a moment. Seems weird to have all this board space and not enough.
Did you say in the major calyx, that's where filtrate becomes first?
The minor calyx. So two minor calyces are required to form a major calyx. Let's see if I have darker black here. So there is a capsule around the kidney. That's about the last we say about it. There's a capsule present. And drawing backwards, so leaving the kidney, I have this tilted just to give me more room. All right, it's actually going to be more vertical in your body, so this is an artistic license here. Leaving the kidney, we have a structure that is dilated at first and then tapers. Let me turn the light on so we have, actually, do you mind putting the light back on again? This is known as the pelvis. Just touch it once and they'll come on. There you go. Thanks. And then it's going to form the ureter, or ureter, which travels also retroperitoneal down to the empty end of the bladder. So we'll deal with that in a couple of weeks. Draining into the pelvis are channels that are formed by 2 or 3 other smaller channels. So this would be identified as a major calyx. And it's formed by 2 or more minor calyces. So calyces is plural. Now, fitting into each of these, and I'll come over here and draw one a little bit larger. We have a pyramid. All right. Here is a minor calyx, and the tip of that minor calyx looks like a cup. It's indented, it's open. Okay. Fitting into that cup is a pyramidal structure, so it's got a wide base and narrows to a tip that fits into and empties into this minor calyx. Let me bring this actually around here a little bit like that. So this is known as a medullary or a renal pyramid. Sometimes the term renal is used to distinguish it from the brainstem medullary pyramid. So if I were to diagram that over here and just put in 1, that, okay, and I'll put it in. And this appears striated. So even in a whole kidney, that is dissected, it appears to be formed of vertical structures. And it is. So if we were to draw a dotted line, this is not normally found in the kidney. The region outward, superficial if you would, to here is identified as the cortex. And then this is identified